Good evening. It's an honor to be here to share with you my progress in my thesis project. The title, Transcription Regulation of CFTR by cis regulatory elements specific to intestinal epithelial cells. So first, I bring you the plan for today's talk. I'm going to talk about the significance of my research, then the background data, and then moving to each aims, and then I'll give you a summary. So first of all, cystic fibrosis is a monogenetic disease that affects a number of people, 30,000 in the U.S. This phenotype is across the body, different kind of epithelial cells in different organs. Here, my research focus is on the intestinal phenotype of the cystic fibrosis, which first represents as Meconium ileus. It's somewhat debatable, is it pancreas phenotype or intestinal phenotype? But the second phenotype, constipation, chronic constipations, are intestinal phenotypes. There is something wrong with patients' epithelial cells. So one of the major reasons for that is the cystic fibrosis transmembrane conductance regulator, CFTR. It's a rather large protein. It's the ABC class transporter protein that regulates the chloride ion transportation across the membrane. As you can see here, these are the chloride ions that are transported across by CFTR, and mutations in the CFTR causes cystic fibrosis. You realize that I'm a little ambiguous about mutations in CFTR because this is a really complex definition of what causes CFTR, cystic fibrosis. They're all different kind of mutations, either in the coding region or non-coding region, in the intergenic region of this large gene that spans 250 kb region. And here is showing a representative traditional definition of 5 classes of CFTR mutations. First, it can affect the at the nucleus region that has no synthesis of CFTR protein at all. Second class is blocked in the processing, protein folding, and trafficking. One of the famous ones, Del 508, is this class. Next class are gating or conductance mutations represented in the membrane. So these proteins get transported into the correct locations, but they cannot fully function as their roles. And the last case is reduced in synthesis. So there's protein's fine, it doesn't have enough amount in the membrane. So, as I just said, there's so many different kinds of mutations. There's roughly 2,000 different kinds of mutations spanning the CFTR locus. Here I'm showing a representative 47 mutations that are most studied and most frequently. Out of the 312 causal mutations of that 2,000 mutations. And here the allele frequency of these mutations roughly follow the Poisson distribution. There's highly variable in the mutations, then addressing my research, which is focusing on transcription regulation of CFTR, how that is going to help the patients. So right now the current treatment of the CFTR is basically these main players here. The Aficamten, Lumacaftor, and 2018 the FDA has approved Tezacaftor. Both these small molecules address either the protein gating, opening and closing properties, or the trafficking and folding properties. And as I remind you here, the different class mutations happen in different layers of the CFTR. So they applied these drugs into all different classes here, and these 4 classes are the major classes of CFTR. So basically 95 of the patients right now at least have some sort of treatment. And my research, or my proposal here, is to propose transcription regulation of CFTR. By boosting transcription, I can first help all of the existing treatment by providing more transcript to work with. So these protein folding assisters or the gating mutation helpers will have a stronger effect. So first, it's assisting current available treatment. And second of all, we can studying the CFTR transcription will help the people who has transcript but no protein. And those drugs are currently 5% of people that have no treatment. So if we study transcription regulations, ultimately we can figure out a way to treat this people. And then finally, there's this last group of people who we think are novel mutations harbored in their enhancer regions. These are yet to be validated by more research, but our group is on the edge of discovering and validation of these kind of mutations in the enhancer region. So then, moving on to talk about what I'm going to do. So first, I'm going to do is to study the enhancer corporations of CFTR in the intestinal epithelial cell. Second point I'm going to do is bring the previous existing knowledge of the past decade to finally transition it to a organoid model to address towards the treatment finally, using enhancer activation as a potential treatment for CF, a type of CF splicing. So first thing here, before I got into my research, the background of other people have done a lot in this area. So here is a graph showing an open chromatin map of in the Kaggle2 cells. So here representing two enhancers that we have identified, 1 and 11. These enhancers are tissue specific, which addressed in here. So large amount of data has been, people have been done this research in the luciferase assay. Here first is address this intron 1 enhancer. It is active in Kaggle2 cells, which is the intestinal epithelial cells, but inactive near zero activities as control in the 16HB cells, which are lung cell, lung epithelial cell, and HEC393 cells. Here is the intron 11 elements, is zero activity in the lung epithelial cell. Here it shows that in Kaggle2 cells, both the elements by themselves have robust activities, and when people combine them together, it has an even stronger activity, suggesting their potential cooperations. Here is the, so previously people dive into the more detail of the research to show why it's some lag players to bind transcription factors. In the intron 11 region, we have found this robust binding of FOXA2. And intron 1, we have found a robust binding of HNF1. Both of these are transcription factors, and even here, we have shown knockdown of FOXA2 has decreased CFTR transcript levels. Then, in more recent years, 2016, we have adapted 4CSeq to tease apart the architectural regulation of CFTR. So the shown here is a wild-type Kaggle2 cell. It's a promoter as a root point. So as you can see here, these light blue color indicating less interaction frequencies with this promoter. And then the purple color indicating here are more frequently interacting with the promoter. And the red indicating a strong interaction with the promoter. And the way to see this graph, usually along the viewpoint, it makes sense that it has strong interactions because it's close proximity. As you move across further down, we observe intron 11, which is the robust enhancers we previously talked about, has indeed moved back to the promoter regions. And that's what I wanted to use that for. And then addressing to my aim, which is first to delete these enhancers in the Kaggle2 cell natively using CRISPR-Cas. Here the experimental design is using Cas9 protein and a guide RNA transfected into cells, and then sort for GFP positive cells, then single cell expansion of those clones, and then we do genomic DNA and PCR validation and single sequencing. So luckily, I obtained the clones of deletion of intron 1. Previously in the lab, people had generated deletion of 11. Then the qPCR assay of CFTR shows a 30% reduction of transcript after removing of intron 1, and about 80% reduction in intron 11. So next, after obtaining these kind of data, I will move on to deleting both enhancers in the same together to investigate if these expression levels will further drop down. And also I will be investigating if the transcription factor occupancy changes after deletion one enhancers if the other enhancers increase or decrease enhancer activities, suggesting their response in compensating the other enhancer absence. And also the other, from the other side, our address is to use enhancer inactivation or repression to as a duplicate validation of my deletion of enhancers. So we can use this B-Cas9 crab and SID complex, which crab induces heterochromatin formation. SID induces similar response. So here I showed my design of this 5' RNA covering the intron 11 regions. I have other 5' RNA covering the intron 1 regions. And after inducing transfecting these RNAs and plasmid coding for these proteins into the cell, we are likely to see a similar response of repressing the enhancer with our deletion of the enhancer. Next step, I'm going to move on to editing those enhancers using the CRISPR 2.0 technology. Here I'm showing a intron 11 FOXA2 binding site I just showed previously. Here's the guide RNA design. The A4 region will mutate it from A to C site. And since these sequences are the core binding site of FOXA2, mutation of a single base pair are predicted to have a dramatic effect in the binding affinity of FOXA2, both locally and the enhancer 1.5 kb region as globally will have a dramatic effect. And here we predicted that after induce this enhancer mutation, we are likely to see FOXA2 binding decrease in this specific site. Also, since this intron 111 is very important for the transcription regulation of CFTR, we're likely to see decrease or reduction in CFTR expressions and loss of the enhancer activity, since we believe that FOXA2 is one of the drivers that open up this chromatin and maintain open chromatin and have the enhancer activity. And the next thing is to study the splicing mutation in the organoid model. So first I would like to address this mutation, which is occurred in 1.2% of the CF patients, is have a mild phenotype compared with the general CF mutations. It has the sweat test is mild, and also the pancreatic insufficiency frequency is lower. This mutation is the most frequent ones that currently have treatment that are splicing mutations. I'll come again slowly. So the splicing mutations, the most common one that have treatment that are splicing mutations. The reason we chose splicing mutation for this project is all of the other mutations that are known to cause CF are protein coding mutations or folding or other things. The splicing mutations are closely connected to transcription, which is the subject study. That will be a good candidate to test our hypothesis. Here is a molecular mechanism of why this mutation causes CF. The mutations here cause a aberrant splicing. And these transcripts have this introducing this aberrant sequence, and this sequence harbor stop codon, and they have no protein produced. And also this splice will produce also a normal transcript and this transcript. So both of these are existent. And the current treatment for this mutation is using ivacaftor, which come to you as somewhat surprising. If you remember previously, the ivacaftor is a small molecule that helps the gating mechanism of the CFTR protein. So this is splicing mutation, why does gating help? There have been clinical trials that shows that giving this drug, the existing CFTR protein in the membrane will tend to open, so that compensated the loss of less amount of normal CFTR in here. So, but keep in mind that's not the addressing the problem. It's more addressing something downstream to help out with this process. So what we propose here is to use this enhancer activation VPR system. I will remove this part later on. The VPR system will be used to override the transcription in this slide here. We will increase the transcription. So we will both increase the normal transcript and the variant transcript compared to the wild type here, or the control group. So this way, we will force the cell to pump more functional CFTR into the membrane, and then this future could be even co-treated with the abacavir as a combination treatment to show if the combination treatment shows effects. Then I propose the functional readout of these assays to be the following. So first, assay the RNA level by the qPCR and the protein level by Western blot. And since we are doing the organoid, there's a protocol developed by Jeff Beekman's lab that we could use stain by this calcine. It's a screen stain, and then use forskolin as a specific inducer of CFTR. Forskolin induces CFTR channel to be open. So as the wild type here, there is after induced by forskolin, the green area increases, but the CFTR now has not changed at all. And here's this example data they have in the paper shows that the normal and the mild CF patient and the severe CF patient has basically zero CFTR function, and this will have somewhat conserved CFTR function. So that's the plan for A2, and in summary, my research first will, in one, will address the CFTR enhancer cooperations in the intestinal epithelial cells. Second, I will propose to expand our understanding in the cell lines to an organoid model to address the potential treatment for the splicing mutations. And thank you for listening. We have a appreciation slide here. Yes, thank you very much.